Hello, my dear friends. Today, let us talk about the plant tissue culture, its definition, media, steps, types, and plant tissue culture uses. Plant tissue culture is the in vitro aseptic culture of cells, tissues, or whole plants under controlled nutritional and environmental conditions, often to produce clones of plants. This technique primarily relies on plant cell totipotency, which is the capacity of a single cell to express the whole genome during cell division. The ability of cells to change their metabolism, growth, and development is just as significant and essential for the regeneration of the entire plant. Plant tissue culture technology is being widely used for large scale plant multiplication. In addition to being used in research, they are now essential for plant propagation, disease eradication, and the generation of secondary metabolites. Now, let us see about the plant tissue culture theory. A non scientist, Gottlieb Haberland, in 1902 tried to cultivate individual pellicide cells from leaves in Knopp salt solution supplemented with sucrose. The cells sustained for a month stored starch but ultimately did not divide. Despite his failure and features, he is considered the father of plant tissue culture since his experiment set the stage for developing tissue culture technology. Similarly, Roger J. Gautret, a French scientist, had encouraging results with culturing cambial tissues of carrot in 1934. Now let us understand about the conditions for plant tissue culture. The choice of medium is based on the types of plant species. Explants are used for culture for optimal responses. All the nutrients required for a plant's proper growth and development should be present in the plant tissue culture media. Macronutrients, micronutrients, vitamins, and other ingredients, plant growth regulators, a carbon source, and in case of a solid medium, a few gelling agents make up the majority of its composition. Similarly, hormone levels and culture variables like temperature, pH, light intensity, and humidity play an important role in the success of tissue culture. The minerals consist of macronutrients such as nitrogen, potassium, phosphorus, calcium, magnesium, and sulfur, and micronutrients such as iron, manganese, zinc, boron, copper, molybdenum, and cobalt. Vitamins are necessary for the healthy growth of plant tissue cultures. The vitamins like thiamine, vitamin B1, pyridoxine B6, and nicotinic acid niacin. Other vitamins such as biotin, folic acid, ascorbic acid, vitamin C, and vitamin A tocopherol are sometimes added to the media formulations. Plants also require an external carbon source. Sugar, the most commonly used carbon source, is sucrose. Other sources used are glucose, maltose, and sorbitol. The pH of the culture medium remains vital as it influences the uptake of various components of the medium and regulates a wide range of biochemical reactions. Most media are adjusted to a pH of 5.2 to 5.8. A higher pH may be required for certain cultures. Now let us understand about the plant tissue culture media. The most popular medium for in vitro vegetation propagation of various plants is Moshes and Skoog medium, also known as the MS medium. For culturing either a solid or liquid medium can be employed. Next is the McKeown's woody plant medium. WPM has been widely used for tree tissue culture. Next is the Kunson's medium is used for the orchid tissue culture and fern tissue culture. Now, let us understand about the plant tissue culture growth regulators. Plant growth regulators are crucial for determining the development of plant cells and tissues in a culture medium. The most commonly used plant growth regulators are auxins, cytokines, and gibberellins. High oxygen concentration often favors the development of roots. The most commonly used oxines are IAA, indole acetic acid, IBA, indole butyric acid, and NAA, naphthalenic acetic acid, and 2,4-D means 2,4-dichlorophenoxyacetic acid. Cytokines promote cell division and shoot growth. The most commonly used cytokines are BAP, benzyl aminopurine, zeatin, isopentyl adenine, 2IP, and the kinetin. Cytokines are generally dissolved in dilute HCl or NaOH. A clump of undifferentiated cells called a callus develop when auxin and cytokines levels are balanced. Now understand about the vessels. Another critical aspect in plant tissue culture is the management of gaseous hormone ethylene. In closed culture vessels used for in vitro plant growth, ethylene builds up and is often detrimental to the cultures. The addition of ethylene biosynthetic inhibitors such as silver nitrate, AVG, aminoethoxy, vinylglycerin, and silver thiosulfate have been shown to increase the formation of shoots. Cultures are grown in walk-in growth rooms or growth chambers. Humidity, light, and temperature must be controlled for the proper growth of cultures. A 16-hour light photo period is optimal for tissue culture, and a temperature of 22 to 25 degrees Celsius is used in most laboratories. Cool white fluorescent lamps also supply a light intensity of 25 to 50 micromole meter per second. Relative humidity of 50 to 60% is maintained in the growth chambers. Some cultures are also incubated in the dark. Cultures can be cultivated in various containers, including test tubes, flasks, petri dishes, and bottles. Now, let us see about the sterility. The preservation of a sterile environment is necessary for effective tissue culture. The laminar flow hood is used for all tissue culture work. A dust filter and a high efficiency particulate air HEPA filters are used in the laminar flow hood to filter the air. The hood must kept spotless, which one can be accomplished by wiping it with alcohol that contains 70% of alcohol. The surfaces of plant tissues naturally contain a variety of bacteria and fungi. Before tissue culture, it is crucial to thoroughly clean the explant since contaminants can proliferate in the media culture. They also compete with the plant tissue for nutrients, depriving it of those nutrients. Fungi and bacteria can rapidly surpass plant tissues and destroy them. Explants are commonly surface sterilized using sodium hypochlorite, ethanol, and fungicides when using field grown tissues. The type of tissue usually decides the time of sterilization. Leaf tissue requires a shorter sterilization time than seeds with a hard seed coat. Now let us understand about the types of plant tissue culture. First one is the callus culture. A callus is an unorganized mass of cells that develops when cells are wounded. When the explant is cultivated on media that promote the development of undifferentiated cells, a callus is formed. The majority of callus cells are formed with the aid of auxins and cytokines. Using plant growth hormones, callus can multiply continuously or be directed to develop organs or somatic embryos. Now let us understand about the second type of the plant tissue culture, that is the cell suspension culture. Small fragments of loose friable callus can be cultured as cell suspension cultures in a liquid medium. Cell suspensions can be maintained as batch cultures grown in flasks for long periods. A portion of callus tissue can be transformed into a liquid medium, and when subjected to continuous shaking, single cell cultures and suspension cultures can be cultivated from callus cultures. The growth rate of the suspension cultured cells is generally higher than that of the solid culture. Next type of culture is the anther or the microspore culture. The culture of anthers or isolated microspores to produce haploid plants is known as anther or microspore culture. Embryos can be produced via a callus phase or be a direct recapitulation of the developmental stages characteristic of zygotic embryos. Compared to the traditional breeding methods, microspore culture enables the creation of homozygous plants in a very short time. These homozygous plants are useful tools in plant breeding and genetic studies. Now, next is about the protoplast culture. Protoplasts contain all the components of a plant cell except for the cell wall. Protoplasts can be used to create somatic hybrids and regenerate whole plant from a single cell. Cell walls of X plant can be removed either mechanically or enzymatically. Protoplasts can be cultured either in liquid or the solid medium. Protoplasts embedded in an alignment matrix and then cultured on a solid medium have better success rates of regeneration. Although protoplasts appear to be a very appealing method for regenerating plants and transferring genes that are extremely delicate. Next about the embryo culture. It is a technique in which isolated embryo from immature ovules or seeds are cultured in vitro. For species, both seeds are dormant, resistant, and prematurely sterile. Embryo culture has been used as a helpful tool for direct regeneration. In plant breeding programs, embryo culture goes hand in hand with in vitro control of pollination and fertilization to ensure hybrid production. In addition, direct somatic embryos and embryogenic calyces can be produced from immature embryos. Next is about the meristem culture. Using apical meristem tips, it is possible to produce disease-free plants. This technique can be referred to as meristem culture, meristem tip culture, or the shoot tip culture, depending on the actual explant used. Plant apical meristems makes good explant for cultivation of virus-free plants. Hence, this method is usually used to eliminate viruses in many species. I hope it will be understandable to all of you. Now let us talk about the regeneration method of plant tissue culture. It includes two types of method. One is organogenesis and next is the somatic embryogenesis. Now let us see a small introduction about both. Organogenesis in plant tissue culture, it refers to the formation of either shoot or root. The equilibrium of auxin and cytokinin and the tissue's capacity to react to phytohormones during culture as the key factors is in vitro organogenesis. In vitro organogenesis can be of two types, indirect organogenesis and the direct organogenesis. Direct organogenesis involves direct bud or shoots formation from the tissue without a catastasis. Plants are usually propagated by direct organogenesis for improved multiplication rates and production of transgenic plants, but mainly for clonal propagation. Now let us talk about the indirect organogenesis. Involves the formation of organs indirectly via a callus phase for the production of transgenic plant. Induction of plants through a callus phase has been used. Either the callus is transferred, plants are regenerated, or the primary explant is transferred, and the callus is formed and shoot are cultivated from the explant. It is more important for transgenic plant reproduction. Now let us talk about the somatic embryogenesis. Somatic embryogenesis is a non-sexual developmental process that produces a bipolar embryo with the closed vascular system from the somatic tissues of a plant. It has become one of the most powerful techniques in plant tissue culture for mass clonal propagation. So, somatic embryogenesis may occur directly or via a callus phase. For clonal propagation, direct somatic embryogenesis is preferred since there is less chance of somaclonal mutation. Indirect somatic embryogenesis is usually used in the selection of desired somaclonal variants and for the production of transgenic plants. Encapsulated somatic embryos are known as the synthetic seeds. Synthetic seeds have multiple advantages. They are easy to handle, can be potentially be stored for a long time, and there is potential for scale up and low cost of production. Now let us understand about the rooting of shoots. The success of acclimatization of a plantlet directly depend on the root system. Roots of shoots Rooting of shoots can be achieved in vitro or the ex vitro. Ex vitro rooting involves pre-treating the shoots with phenols, oxime, then planting them directly in soil under high humidity, which significantly lowers the cost of manufacturing. This technique also allows simultaneous accumulation of rooted shoots. In vitro rooting consists of rooting the plants in exogenic conditions. Despite the cost factor in vitro rooting is still a common practice in many plant species. Several factors are known to affect the rooting. The most important factor is the action of endogenous and exogenous auxins. Phenolic compounds are also known to have a stimulatory effect on rooting. Uh, Phylloxera, a root promoter, is reported effective in root development. Catechol, a strong reducing agent, has been reported to regulate IAA oxidation. Now let us talk about the acclimation or the acclimatization. Once plants are generated by tissue culture, they have to be transferred to the greenhouse or the field. This requires that the plant be hardened off before transfer to the field. To reduce water loss during acclimatization, plants are initially transferred to a greenhouse or a growth chamber. The relative humidity outside the vessels is often significantly lower than the humidity inside the vessels. Once the plants are acclimatized under the greenhouse conditions, they are ready for transfer to the field. Now let us talk about the advantages of plant tissue culture. Totipotency, nutrition, metabolism, division, differentiation and preservation of plant cells. Morphogenesis and plant regeneration from individual cells or tissues through organogenesis or the somatic embryogenesis. Variations were generated through in vitro culture. Evolution of haplite through anther and pollen culture, including the ovule culture, wide hybridization programs through ovule, ovary, embryo culture to overcome both pre-zygotic and the post-zygotic sterility mechanisms, micropropagation of plant materials, in vitro selection of mutants tolerant to biotic and abiotic stresses, in vitro culture and secondary metabolite symbiosis, plant genetic engineering using DNA transfer, and in vitro culture techniques. So it's a very useful technique. The technique is the plant tissue culturing via taking explant from any parts of the plant as we had studied. Okay guys, thank you so much. So like, subscribe your own YouTube channel, that is Biotic World. Be a member of our family of Biotic World.